Good morning. I'd like to, begin with my disclosures. I do have some connections with various companies, advisory panels, etc., but there's nothing in terms of my connections with these companies that I'm going to be discussing in this lecture this morning. In the late 1980s, early 1990s, a large project was undertaken called the Human Genome Project. And with this project, the goal was to develop the adenine, thymine, guanine, cytosine sequence of the human genome. And it took 13 years before we were able to crack this human genome code. And here we see in this slide two of the journals from which that were published in 2003 that showed the human genome. One was done by the private sector, and the other one was done by the public sector. And in Time magazine, the two major scientists that were involved with this project were recognized for this work. With this development of the human genome, all kinds of new technologies were developed, including the ability to do better and faster sequencing of DNA. New statistical techniques were developed. Bioinformatics were developed, and some investigators began to look at this in terms of maybe taking this technology and beginning to look at sequences of microbes in our body, sequences in the soil, sequences of DNA in the oceans. And so they began to find that using culture-based techniques, we were only able to scrape the very tip of the iceberg in terms of the numerous quantities of microbes that are living with us in this world. And one of the areas involved the human gastrointestinal tract. And with this, these studies in the human gastrointestinal tract, it was found that only about 30% of the microbes in the human gastrointestinal tract could be cultured. And so with this, the National Institutes of Health decided to undergo this human microbiome project where the major goals were determining whether individuals share a core human microbiome. In other words, the microbes in the intestine, are they the same from one individual to the next? They wanted to understand whether changes in the human microbiome can be correlated with changes in human health. They wanted to develop new technological and bioinformatic tools needed to support these goals, and they also wanted to address the ethical, legal, and social implications raised by the human microbiome research. There are many non-culture-based technologies, and I'm not going to get into any of the details of these, but some of them are fairly simple. At the top, you can see some gel separation techniques, which are quite simple, but nowhere near as detailed as some of the techniques that you can see further to the bottom of this slide. Shotgun metagenomics, which actually takes a whole microbial colony in a system and looks at all of the DNA in that particular system. And also bioinformatics and computational methods, and this new area called systems biology is being utilized in these non-culture-based technologies. Well, here I just want to show you with trying to culture the microbes in the human gastrointestinal tract, if we work really hard to try to culture all the microbes in the gastrointestinal tract, we might be able to see 400 species. But using some of these new DNA-based technologies, it is thought that we are going to find approximately 2,000 operational taxonomic units or species of microbes in our gastrointestinal tract. When we look at the microbes in the gastrointestinal tract, there's an aboral gradient from the top to the bottom. In the top of the gastrointestinal tract, for example, the stomach, you see very few microbes, approximately 10 to the 1 or 10 to the 2 microbial organisms per milliliter of fluid. But as you go down further in the gastrointestinal tract, into the ileum and into the colon, you start seeing huge numbers of microbes, to the order of 10 to the 12th, or trillions of microbes. So what I want to talk about over the next several minutes, I want to just give a little background of the microbial ecology of the developing gastrointestinal tract, talk a little bit about the fetal microbiome, the C-section versus vaginal deliveries and subsequent outcomes, a disease that we see often in preterm babies called necrotizing enterocolitis, talk a little bit about the implications of early and frequent antibiotic usage, and also the autoimmune microbiome, talking a little bit about type 1 diabetes and some of the challenges that we have ahead of us. When we think about the microbes in the gastrointestinal tract, we usually think of the bad and the ugly. We think that these microbes cause diseases. Well, we're just beginning to understand that the microbes that live in our gastrointestinal tract are very important. They serve many important functions, and without them, we have a major problem in terms of health. Here are just some of the major functions of these commensal microbes that live within our gastrointestinal tract. They're important for nutrient metabolism. For example, vitamin K production. Other vitamins are also dependent on microbes in our gastrointestinal tract. They produce short-chain fatty acids, which are very important for growth of the colonic epithelial cells. They're important for tissue development, resistance to colonization with pathogens. They maintain intestinal homeostasis, and now we're beginning to find that there are some studies that show that they are important for development of the brain. And the type of microbes we have in our gastrointestinal tract as babies may actually result in our behavioral differences when these individuals grow older. We have two major functions that I'd like to separate the functions of the intestinal microbial system into. One is called the bioreactor role, and the other is called the immunologic role. The metabolomic or the bioreactor role can be seen on this slide. If you look at the top, you see this animal called a termite. And they look a lot like ants, and as you know, termites will eat wood. And these termites have certain enzymes in their gastrointestinal tract called lignases. Lignases will break down very complex carbohydrates, such as found in wood. So these animals can actually survive on eating something like wood. We do not have those types of enzymes in our gastrointestinal tract. However, we do have certain microbes that are able to digest very complex carbohydrates, perhaps not as complex as what we find in wood, but complex carbohydrates. And the efficiency of our digestion of these carbohydrates may actually end up making us obese or thin. And there are several studies now showing that the types of microbes in the gastrointestinal tract may actually play a role in obesity and non-obesity. The other major role outside of the bioreactor role is the immunologic role. This is from some work that was done in the early 1960s at the University of Chicago, where they looked at baby rats who were raised in a germ-free versus a conventional environment, and they took out the axillary lymph nodes from those animals. And at the top of the slide, you can see an axillary lymph node from an animal that was raised in a conventional environment. In other words, conventional, exposed to the outside microbes. And you can see that this is a very normal looking lymph node. At the bottom, you see an animal that was raised in a germ-free environment. And you can see that this is very different in structure compared to the conventional raised animal. So it is important for us to be exposed to microbes during our early development. Now, when we try to do studies in humans and development of the microbial ecology of the gastrointestinal tract, our major access is to be able to get fecal samples, poop samples from babies. And so work that is done in our laboratory and other places where they are doing this type of work, you have to take a lot of diaper samples and poop samples, and you have to save them in freezers. And at this juncture, we have 3 freezers full of material from babies involved in our study. Now, what about some of the other studies that have been done that have looked at the development of the intestinal microbiota? One of the first studies was done at Stanford University by Palmer and colleagues, where they looked at 14 healthy, full-term infants. And some of these were born by vaginal delivery, some were born by cesarean section. And they used a relatively uncomplicated, unsophisticated technique that actually looks at specific microbial DNA sequences. And they followed these babies for their entire first year of life. Now, this looks like a very complex slide, but it shows the microbial DNA concentration of these babies over the first year of life. And so what you can see from these figures is that some of these babies, right at the very beginning, for example, this baby here, had microbes in the meconium. Over here, this baby had microbes in the meconium. Another one over here, microbes in the meconium. It's a dogma. It's a well-known supposed fact that the baby's first stools do not have microbes. And so in this study, they were actually seeing microbial DNA in these babies' first stools. Another finding you can see if you look at some of the arrows here, the blue arrows, you can see that there were drops in the microbial DNA. This was a time when these babies were getting antibiotics. And so you can see that the microbial DNA drops markedly at the time of antibiotic usage. I'll get back to this a little bit more later. Another study that was done in one baby, one term healthy baby, and studied over 2.5 years, and actually reported in one of the most prestigious journals of the world, the PNAS. This was almost unheard of to have a study published on one individual. But this is a very important study because this study longitudinally looks at one baby over a period of 2.5 years. And it's thought that probably one of the authors was the mother of this particular baby, because otherwise it probably could not have been done. So what happens? You look at this baby in terms of the early concentrations or the early microbes. If you look at the mother, you see a very high diversity of microbes. If you look at the meconium, you see a lot lower diversity, but you still see microbial DNA. Another very interesting finding, just like the last study. If you go down a little bit further, you see that the diversity of the microbes begins to increase over time, but then there's a slight drop. Not that terribly evident over here, but this is when the baby received his first dose of antibiotics, and over here, another drop. More significant drop when the baby received the second dose of antibiotics. Then they looked at this much more closely in terms of the types of microbes. And here, I don't want you to focus on all these dashes here, but I'd like you to see up here, there are 4 major phyla of bacteria in our gastrointestinal tract, and they found these 4 major phyla of bacteria in this particular baby's gastrointestinal tract. And then you look at the bottom. These are the colors that represent the different phyla. And here we see that very early on in the first several months, we see primarily the proteobacteria and the firmicute phyla. And then as soon as the baby starts getting rice cereal, table food, and other types of intakes, you start seeing other colors become incorporated. So other bacteria are beginning to develop in association with the changes in feeding in this baby. Now, before I mentioned that this dogma about the mammalian fetal intestine, that it's thought to be essentially sterile, and the first exposure to maternal microbiota occurs during passage through the birth canal during the first hours of delivery. Well, we are beginning to find that that's really not the case. Microbes are able to ascend from the vagina into the amniotic cavity through translocation through the chorioamniotic membranes. And during the last trimester of pregnancy, the fetus swallows approximately 400 to 500 milliliters of amniotic fluid per day. And with this swallowing of the amniotic fluid, the fetus is also swallowing a large quantity of microbes. So it's not terribly surprising to find microbes or microbial DNA in the meconium of these babies early on. Now, why is this important? Well, it's thought that perhaps the microbes in the amniotic fluid could actually contribute to the production of prematurity. This is work that was done at Dr. Roberto Romero's laboratory, and Dr. Roberto Romero has large quantities of amniotic fluid from different mothers that date back to the 1980s in his laboratory. And these amniotic fluid samples are from mothers who delivered prematurely or delivered close to term. And so one of the things he did was he took the amniotic fluid and looked at the microbial DNA content of amniotic fluid at different gestational ages. And these are mothers who did not have chorioamnionitis, at least not diagnosed chorioamnionitis. And here at the bottom, we see the bacterial DNA. And in the y-axis, we see gestational age at delivery. And here we see that there's an inverse relationship between these two. So the more microbial DNA, the earlier that baby is born. So in other words, if you see a baby with amniotic fluid gene copies of 10 to the 6th or 10 to the 7th, that baby is more likely to be born at 25, 26 weeks gestation. That was also associated, the higher microbial content in the amniotic fluid was also associated with a higher white blood cell count and also interleukin-6 level, and this suggests more inflammation, and we know that the inflammatory response is one that can trigger premature labor. A lot of work still needs to be done in this particular area. Work that was done in our laboratory looking at meconium analysis, looking at the diversity of the Meconium microbes was very interesting in this regard. Here we see the Meconium in mothers who were treated with antibiotics versus no antibiotics, and we see a marked difference in the diversity of the microbes. Gestational age, babies born later had a lower diversity, a higher diversity than those babies born earlier. And there was also a difference in the diversity of the microbial ecology of the gastrointestinal tract depending on what these mothers intended to feed their babies with. Not necessarily that the babies were even being fed breast milk versus formula, but just the intention of breastfeeding versus formula seemed to show a difference. Why? Perhaps because of the socioeconomic level. In our hospitals, we see that mothers with higher socioeconomic levels tend to breastfeed their babies considerably more than those babies at the lower socioeconomic levels. And we know that there's a different microbial ecology with mothers of different socioeconomic levels. What about vaginal delivery versus C-section delivery? Well, currently in the United States, the C-section delivery rate is around 33%. 20 years ago, it was only about 15%, so it has gone up fairly markedly. But that's actually relatively low in comparison to some other countries. In China now, the C-section rate is about 60%. In some other countries, for example, Brazil, the C-section rate is up to 90%. Does this pose a public health problem in terms of the microbial ecology and what happens to these babies later on? Well, with the vaginal delivery, there's exposure to vaginal microflora and there's supposed normal microbial seeding of the gastrointestinal tract. And the first year of a baby's life is the most important in terms of development of the immune system. What if you have a C-section delivery and the baby is not exposed to the normal vaginal microflora? There could be potentially abnormal development of immunity. A study that was done in Venezuela looking at non-culture based techniques to look at the microbes on the baby's skin and in the baby's mother's skin and the microbes in the baby's gastrointestinal tract showed that those babies who were born by vaginal delivery, seen in the red, seen in the red on this slide, in the pink, those seem to cluster very closely together. Whereas those babies who were born by C-section delivery, seen in the blue over here, seem to cluster much more to the mother's skin microflora rather than her vaginal microflora. So there's a real difference in the development of the microbes in the gastrointestinal tract of these babies. Why is this important? Well, epidemiologic studies have shown that babies born by cesarean section have a higher incidence of celiac disease, type 1 diabetes, gastroenteritis and asthma. So could it be that the microbial colonization in the gastrointestinal tract could lead to these differences in these diseases? Here's a disease that we see a lot in the neonatal intensive care unit. This is a preterm baby with necrotizing enterocolitis. Very, very important problem. It occurs in about 7% of preterm babies in the United States and Canada, and about similar, maybe a little bit less throughout Europe. Very important disease. What happens to these babies? Well, a lot of these babies, most of our preterm babies, are treated with antibiotics. The 10 most commonly used drugs in the neonatal intensive care unit are seen here, and at the top of this list are ampicillin and gentamicin. 90 to 95% of preterm babies get treated with antibiotics shortly after birth. Could this have an effect? Well, in terms of necrotizing enterocolitis, a very recent study looking at days on antibiotics and the odds ratio for the development of necrotizing enterocolitis suggested a higher or an increasing rate of necrotizing enterocolitis with more use of antibiotics. We are also doing a study at the University of Florida that involves looking at preterm babies and the development of necrotizing enterocolitis and relating that to the microbes in their gastrointestinal tract. We are taking babies and collecting stool from all of these babies while they are hospitalized, and if they develop necrotizing enterocolitis, we take a closely matched control baby and look at the microbes and compare the microbial ecology of those two groups of babies. And this is from a recent publication in PLOS One, came out last May, looking at cases versus controls. And there's several interesting things on this slide, but I don't have time to go into all of the details. But we see that those babies who were the cases, the necrotizing enterocolitis cases, if you look at their antibiotic usage, each one of these letters under antibiotics represents antibiotic usage. You see a lot more letters in the cases of necrotizing enterocolitis compared to the controls. So again, there appears to be this relationship between use of antibiotics and necrotizing enterocolitis. The types of microbes that we see prior to the development of necrotizing enterocolitis are seen in this slide. And if you look at the controls 1 week before the diagnosis, or cases 1 week before diagnosis, these colors represent the different phyla, and we see that there's a real change 1 week before the diagnosis in the cases to 72 hours, within 72 hours of diagnosis. You see this marked shift in the firmicute to proteobacter ratio, which you don't see in the control babies. When you look very closely and dig a lot deeper into the microbial ecology, if you look at sequencing on a genus and species level, we look at the cases 1 week before, and here on the dotted lines you see a certain area of this heat map that looks at certain types of microbial operational taxonomic units, and 1 week before we see none. But then within 72 hours, the same area, we start seeing some development of microbes in the necrotizing enterocolitis cases. And what this is telling us is that there are certain microbes that we see in before the development of necrotizing enterocolitis that could be potential candidates for pathogens for the development of this disease. We still have a long ways to go to identify these, because these are microbes at this juncture that don't even have a name. They only have DNA sequences. Now, I'd like to just shift gears a little bit and talk about what has happened over the last 50 years in the relationship between infection, antibiotic usage, and diseases. Over the last 50 years, we see that there's a marked difference in the incidence of infectious diseases. Rheumatic fever, hepatitis, mumps, measles, TB have markedly decreased over the last 50 years. Our usage of antibiotics has also increased over this last 50 years. Our usage of antibiotics in the food that we eat in agriculture has also markedly increased over these last 50 years. What are we seeing over these last 50 years? In addition to this decrease in infectious diseases, we are seeing an increase in the incidence of certain immune disorders such as type 1 diabetes, multiple sclerosis, Crohn's disease, and asthma. Well, this could be related to something called the hygiene hypothesis, and there are certain countries where the hygienic conditions may not be quite as good as other countries. Usually the poorer countries tend to have worse hygienic conditions. Well, this was looked at by Jean-Francois Bach and reported in the New England Journal of Medicine in 2002. And here we see countries with a higher per capita income, greater than 18,000, equivalent in US dollars, versus less than 14,000. And the incidence of multiple sclerosis, childhood diabetes, and asthma are all lower in the poorer countries. We don't really know the exact mechanism of this, but it's thought that perhaps the hygienic system might be too hygienic in some of these more developed or richer countries. And this actually plays a role in terms of the types of microbes that are found in the gastrointestinal tract. Here we have a report from 5 countries looking at the types of microbes using non-culture-based techniques in these 5 different countries, and we see that there's a clustering. For example, here in Stockholm, we see a clustering of the microbes in this particular area of this principal component analysis plot. We see other countries, for example, Germany over here, clustering in this area. So there's differences in the countries and the types of microbes that we see in the gastrointestinal tract in different countries. Could this be important? Well, there are certain countries, for example, Finland, Sweden, Scandinavia has a very, very high incidence of type 1 diabetes. Individuals with a close genetic makeup in Estonia, for example, have a very low incidence of type 1 diabetes compared to those individuals in Finland. So what is it that is causing this difference in the microbial ecology and that is causing this difference in the development of type 1 diabetes, and could they be related? Well, a lot of work is being looked at in this area, and this is some work that was done in our laboratories at the University of Florida, working with Dr. Eric Triplett, who's a microbial ecologist. And here at the bottom of the slide, at the left side, you can see 4 cases and 4 controls. This is individuals who had recently converted to having antibodies for type 1 diabetes and looking at the microbial ecology in their stools. And we see some real differences in the controls versus the cases. And then when you do an analysis of the function of these bacteria, see at the right of the slide, there's a difference in the function of these bacteria. You see that there's a much higher incidence of butyrate-producing bacteria in the control infants compared to the cases. And butyrate is thought to be very important in colonic permeability and colonic function. And so is it possible that what is happening in this situation is seen at the right of the slide? And what we have is the types of microbes producing butyrate or producing substrates for the formation of the barrier, the intestinal barrier, and that potentially allowing for more bacterial translocation or toxin translocation to antigen-producing cells, which then affect undifferentiated T cells to turn into T effector cells versus T regulatory lymphocytes. And could that potentially have an effect on the development of type 1 diabetes? We really don't know the answer to that yet, but this is a very interesting hypothesis. So I'd like to summarize and give you some take-home messages. The intestinal microbial environment during early life may have a major influence in subsequent health and disease. We're just starting, and we have many challenges. For example, what are the best techniques to use for microbial analysis? Just naming the microbes is not going to be enough. We need to correlate these microbes to function. This is going to be critical. New technologies for sequencing, handling of large numbers of samples, large data sets, and statistical analyses need to be developed. And we have to have teams with diverse expertise that need to work together. You have to have clinicians, scientists who are working in the physiologic area. You have to have mathematicians, you have to have statisticians, microbiologists, all working together as teams in this area. Single solo investigators are not going to be able to make big breakthroughs in this area, so you have to have teams. Thank you for your attention.